Hello again, fellow biologists. This is Dr. Tarun, and we are now ready to start with Unit 3, Segment 3, and we will cover the entire chapter of Chapter 11 on cell division, variation, regulation, and cancer. So, this is the first of five video series in this segment. And so this is section 11.1 on cell division. There are five core concepts that we need to cover in chapter 11 on cell division. The first is that during cell division, a single parental cell divides into two daughter cells. Number two, mitotic cell division is the basis of asexual reproduction in unicellular eukaryotes, and also the process by which cells divide in multicellular eukaryotes. Three, meiotic cell division is essential for sexual reproduction, the production of offspring that combines the genetic material from two parents. Four, the cell cycle is regulated so that cell division occurs only at appropriate times and where and when it is needed. Five, cancer is uncontrolled cell division that results from mutations in genes that control cell division. Now, each of these core concepts are going to be focused on in each of the five sections of this chapter. So let's begin with this first core concept, section 11.1. Your reading assignment for section 11.1 is found on pages 225 through 227 in your textbook, although page 227 on cell cycle will actually be covered more fully in section 11.2. The overall learning objective for 11.1 is for you to be able to describe the general process and products of cell division in prokaryotes. The sub objectives for this section is to be able to explain how prokaryotes assure that a copy of their DNA is directed to both cells formed by what we call as binary fission. This is the form of cell division in unicellular prokaryotes. And second is you should be able to explain how cells in prokaryotes divide by binary fission in order to form daughter cells. Again, please don't forget to answer the self-assessment questions. Very important in helping you for the test. Again, let me remind you that it is the self-assessment questions and the learning objectives that I will use in order to formulate the questions for the test. So it really is worth your time to put together answers to these questions. So first is, how is cell division similar and different in prokaryotic cells and eukaryotic cells? Two, how do prokaryotes assure that a copy of their DNA is directed to both cells or to both forming daughter cells from binary fission? And three, what do you predict would be the consequence of a mutation in FTSZ that disrupts the function of the protein it encodes? So this is a special type of protein that we will encounter in the process of cell division or binary fission in prokaryotes. So please pay attention to what its function is. So let's first have an overview of what cell division is. So cell division is simply the process by which cells make more cells. Cell division occurs because of multiple reasons, and the main reason is for cell reproduction or multiplication. And then second is that cell division is also a means by which tissues or communities of cells um, gets replaced during the process of growth and maintenance of the organism, if the organism is a multicellular organism. And it is also involved in repairing damaged cells for tissue healing purposes, and of course, for multicellular organismic growth. So, for example, starting from an egg cell, you end up being a mature organism or mature beautiful individual uh, that has 37 to 40 trillion cells in your body. All of this came about by a process called cell division. Um, so the term cell division is kind of uh, a little bit strange, when in fact what it does is that it multiplies the number of cells in the body. So this is what we call as biological mathematics. So cell division is actually the same as cell multiplication in biology. The process of cell division must satisfy several important requirements. The two basic requirements would be that the two daughter cells must each receive the full complement of genetic material present in the single parent cell. So we need to pay attention on how any type of cell, whether it's a prokaryotic cell or whether it is a eukaryotic cell, what sort of smart mechanism it has in order to ensure that a full complement of genetic material gets transmitted to the two daughter cells after cell division. B, the parent cell must be large enough to divide in two and still contribute sufficient cytoplasmic components to each daughter cell. And so one of the most significant and most incredible phenomenon in cellular life is the ability of the organism to determine the right amount of mass and also the right amount of constituents and organelles in order to ensure that the two daughter cells have all of the supporting mass that they need in order to um, continue to grow as two new daughter cells. So we need to know how these things also happen. In prokaryotic cells, they divide by what we call as binary fission. And in eukaryotic cells, they divide by mitosis and cytokinesis. So these terms might be familiar to many of you, but we will have an opportunity to really understand what they mean in this chapter. So we first look at prokaryotic cells here in section 11.1 and examine the mechanism of binary fission. So in prokaryotes, the binary fission proceeds in 6 basic steps. So the first step is the circular bacterial DNA. Let me see. Sorry. Let me. Right here, this DNA here in prokaryotes is in bacteria is always circular. And this is in contrast to eukaryotes where the DNA is linear. Okay. So you have to remember this. So the circular bacterial DNA molecule is attached by specific protein complexes, as shown here in red, to the inner membrane. So the DNA inside the cell of a prokaryote or a bacterium is actually associated or linked, physically linked to the inner side of the cell membrane of the bacteria, okay, as shown in red. And when binary fission starts, it proceeds first by replicating its DNA. And DNA replication is a very complex process which we will have an opportunity to see in segment 4 of this unit, of Unit 3, when Professor Bosch is going to this lecture on DNA replication. But DNA replication here begins at a very specific location. And what happens is that you have what we will call later on as a replication bubble. If this is the double-stranded DNA, the double-stranded DNA is going to open up in a very specific location called the ORI site. Don't worry too much about this detail yet, but I just want you to have a foretaste of what is happening. So certain groups of proteins are going to open up this specific region in the circular DNA in order to begin replication and proceeds bidirectionally. When it says bidirectionally, it means that this replication bubble here is going to open up this way and that way. So this replication bubble is going to enlarge itself, opening up the single strand for copying itself. Okay, so and this is what's happening in the first step. So a lot of complex biochemical processes are actually happening here, and this happens bidirectionally as I have drawn here on this side. And then third, the newly synthesized DNA molecule, the daughter uh, DNA, which is called newly synthesized DNA, is also attached. And this is very important to note because this will serve as a basis for ensuring that the forming daughter cells are going to have a exact copy of the duplicated DNA. See, so the newly synthesized DNA molecule is attached to the inner membrane near the attachment site of the initial molecule. So let's see what happens next in the next slide. So the fourth step is as replication continues, as it proceeds, the cell starts to elongate. And the elongation process is simply a result of the accumulation, the increase of the components of the cell membrane, as well as the cell wall. By the way, the bacteria always contains cell walls, and they get added into the pre-existing membranes and cell walls, and so they enlarge, but they enlarge in a very specific orientation, meaning the elongation is symmetrical around the midpoint right here. So it goes this way, and it goes that way from the midpoint and of the cell in equal rates. So a very symmetrical elongation pattern. And what it effectively does is that it separates the DNA attachment as they elongate. I think that there is a video in your textbook for this. I will see if I can also attach a video of this to visualize it. But I hope that you can visualize what is happening while the cell is elongating. The DNA attachment sites are separating from each other, which essentially is what is happening. What happens is you are distributing the DNA, the replicated DNA, to two new forming daughter cells. Okay, so this is how you sort out the DNA or ensure that a complete copy of the DNA is distributed to the forming daughter cells. And in number 5, the actual cell division begins. So in 4, we simply have cell elongation. The actual cell division begins with the synthesis of new membrane and wall material at the midpoint. So at this midpoint of the cell, okay, what happens then is the formation of a whole complex of structures, including a ring structure that is made up of protein called FTSZ protein. So the FTSZ proteins form a ring at this site of constriction, and it in fact facilitates the process of constricting the midpoint of the elongated cell in order to form two daughter cells. So these constrictions in between the forming two daughter cells ensures that cell division happens equally and therefore segregating completely separate but duplicate copies of the genetic material or the DNA into the two daughter cells. And finally, this binary step culminates by the uh, continued constriction and separation of the two daughter cells as uh, the FTSZ proteins continue to constrict the separation between the forming daughter cells. So FTSZ protein then is one of the most critical or key proteins that are involved in the formation of these two daughter cells as a result of their constricting function. Okay, so this is essentially how binary fission happens. So at the end of the process, you end up with two daughter cells that have two daughter cells. And not only are they two daughter cells, but they also have similar mass. But most importantly, they have the same DNA molecule inside their cells. And so essentially, these two daughter cells are called clones. They are identical in all respect with each other when it comes to the DNA that they possess. Okay, so this is binary fission in prokaryotes. So to end section 11.1, please do not forget to answer this REEF question. Very easy to answer, low-hanging fruit. Please open your REEF account, and I want to make sure that you answer these questions. Many of you still are, based on what I see on the record, has not been answering the REEF question. So please don't neglect this. This is easy pickings for earning some points for you. Okay, so this should end our section 11.1, and so I shall see you soon in section 11.2. Thank you.